On behalf of, thanks, Bashwat. As I was saying, I'm Dr. Pranita, and I welcome you all on behalf of SINE for this Power Hour Biotech series. Uh, as most of you know, SINE is a technology business incubator hosted by IIT Bombay, and we provide start to scale support to startups, innovators, entrepreneurs through various programs during their journey till scale up commercialization. Uh, today, we are going to talk about one such interesting and very important topic for the healthcare startups, where innovators are engaged in developing new drugs or therapies for various to address the various diseases, disorders related to disorders in India, and we are going to cover the overview of the clinical studies for drugs, which is very important and crucial step to get the approval and commercialize such technology or your product in the India, as well as these studies will also help you in commercializing your product outside India as well. So for this, for such an important session, we have invited Dr. Ravindra Gui, and it's my pleasure to introduce Dr. Ravindra Gui, who is a director at SINE Clinical Services and consultant and trainer in clinical research. Dr. Gui is also holding a position as chairman at various ethics committee. He is also a consultant for BIRAC, and he is an esteemed empanel member for various committees for BIRAC. Dr. Gui has more than 2 decades of experience in clinical research. Previously, Dr. Gui was associated with industry and held prestigious positions at Unichem Laboratories, JK Drugs and Pharmaceuticals, Clinirex Research Private Limited. Post his corporate engagement, he joined academics, and he has held a position of a dean at Bilcare Research Academy of Pune, which is one of the few academics which are dedicated to clinical research education. Dr. Kui also joined then Symbiosis International University as professor for drug discovery and clinical research. Dr. Gui has over 80 papers in national and international journals, co-authored 3 books, and contributed chapters to 5 books. He has done his post-graduation in pharmacology from Karnataka University. He holds PhD degree in medicine from Bombay University and completed his postdoc fellowship at Max Planck Institute for Experimental Medicine, Germany. With this introduction, I welcome Dr. Ravindra Gui for this session, and I request all of you to put your questions in the Q&A chat box. You can as well raise your hand towards the end of the session, where we can unmute you and you can ask your question directly to the speaker and get it addressed. I request Dr. Gui to take this session forward. Over to you, sir.
Thank you, Dr. Pranita, and good afternoon to all of you. Today, we'll be discussing about the clinical studies for new drugs. Normally, this topic is a very, very large subject, and I take it over a period of 10 to 12 hours. Today, I'm going to try to condense it in 1 hour. That's why what I will be able to give you is only an overview. Briefly, I'm going to look at the following parts of it: regulatory clearances, clinical trial designs, controls, blinding randomization, clinical trial sites, inclusion, how we select subjects for that, then the ethical clearance of trials, principles of ethics, phases of clinical trials, adverse effects, and post-marketing studies. These are briefly what I'm going to be speaking about, and then let us see how we will begin with it. First, we must understand what is the meaning of a new drug according to from the legal perspective. Because when we talk about the drug law, the drug regulations, we must know what exactly we mean by a new drug. New drug definition has been broken up into 5 parts. And the first part says any active pharmaceutical ingredient or a phytopharmaceutical derived from plants which has not been used in the country for a significant extent. The second one is a drug which has been approved in the country for certain claims And now it is being proposed for a different claim or different indication, route, dosage form, etc. So the drug is the same, but its usage has changed. That will be considered as a new drug. When you make a fixed dose combination of two or more drugs, now if that combination exists and you change the ratio, then it becomes a new drug. And if that combination does not exist in the market, then it becomes a new drug again. Next is a modified or a novel drug delivery system. Nowadays, you must have read nano drug delivery systems and this type of new ways of delivering the drug. So each time the amount of drug and the rate at which it goes in the body changes. Hence, any modified or a novel drug delivery system will be treated as a new drug. Lastly, a large number of new molecules have come in, which are vaccines, rDNA products, living modified organisms, antibodies, stem cells, and or derived products from stem cells, gene fractions, xenografts. These will all be treated as new drugs simply because these molecules are usually so large that there is no way, no chemical way of saying that a molecule made by you and made by me are identical. Biologically, we have to prove that they are similar and they are called biosimilars, but they'll be always treated as new drugs. Now, all these new drugs must undergo a clinical investigation in the country. And every time before you want to do a clinical investigation, you'll have to get the permission of the drug controller, and that permission application will be made in CT04. And along with that application, you will add certain documents, and these documents are described here. The first is the trial protocol or the clinical investigation plan. Exactly how are you going to investigate it? The proposed informed consent form that every subject who is recruited in the trial must be informed everything of interest to that individual in a form called the informed consent form. And then we take that individual's consent, and that is known as the informed consent form. Finally, case records forms, the forms in which we record the observations, the data that are coming in. There's something called an investigator's brochure. Now, please remember that most of the drugs which we are doing clinical trials are new drugs. They are patented chemicals, and not much information about them will be available either in the books or in the journals or on the net. Hence, we need to provide the investigator with a brochure which contains all the preclinical chemistry and bit of chemistry, actually, preclinical information, whatever we know about its safety, whatever we know about its efficacy, etc., so that the investigator would decide whether to touch that drug or not. Because if you go to an investigator and tell him, I've got a new drug, please use it on your patients, no investigator will try it unless you give all the data that is with you about the drug. The next is something called the investigator's undertaking. It has been prepared by the government. It's a document in which the investigator gives an undertaking that he or she will conduct the trial in the way the government wants it and the way ethically, regulatory way it should be done. And that undertaking is given. The government also wants to know the curriculum vitae of the investigators who are going to be involved in the trials. The government wants to ensure that people who are doing the trials are well qualified. And the government has defined the qualification as someone who is qualified by education, by experience and training. So all these three are required. If at all we plan to use any advertisement to recruit people, to attract people to the trial, then that advertisement material would also be scrutinized by the government. And lastly, the fees. Now, the fees of the government for clinical trials have increased tremendously because the government has to put in a lot of effort before they give you a permission. Currently, for most clinical trials, you have to, sorry. I'm sorry, I just had not switched it off. So the current fees are between 2 lakhs to 3 lakh rupees per clinical trial, and they have to be paid to the government along with the application. Only then the government is going to consider the application. So exactly how do we develop the drugs? We have talked about clinical trials, how to do them. But how do you really develop a drug? First of all, you start with the basic research. From where do you get that drug? Either it is synthesized or it is isolated from a plant, from an animal, from some source. So that is called the basic research, in which you get the molecule. After you get the molecule, you would be interested in knowing that does it really have the biological effect that you want? Every chemical has a biological effect. At least at some dose, they're going to have a biological effect. But that biological effect should be something that you are interested in. For example, if you get a chemical which increases the blood sugar, there's a biological effect. But such a chemical has hardly any application. On the other hand, if you get a chemical which reduces the blood sugar, it may be eventually be useful in the treatment of diabetes. So one has to come to know what effect does it have, and that is what we do in the early discovery, that what sort of biological effect does it have, and does it have the biological effect at a dose which is safe? That is more important. Many compounds will have various effects at unsafe doses. They are not important. What the compound does at a safe level, that is more important. Once this is done, then we go into for preclinical studies. In preclinical studies, we try to find out two things: the safety at different doses and the efficacy of the drug. For this, we may use microorganisms, we may use tissue cultures, cell cultures, we may use different types of cells, we may use animals, animal organs, whole animals also. We might use even animal models of disease. And with all this, we will try to find out two things: that is it safe to use? And secondly, does it produce the biological effect in which we are interested? And once that is proved, then we would go to the clinical development. Now, clinical development is done in usually three phases: phase 1, 2, 3. We'll briefly talk about those later on. And after completing these three phases, if we feel that the drug is worth bringing to the market, an application is made to the drug controller. This is called the NDA application or the BLA application, and the Drug Controller General of India might give you the permission to bring it to the market. And once the drug has come to the market, then we do the post-marketing studies. This entire sequence of events usually takes anywhere between 12 to 15 years for every drug. And currently the costs are put anywhere between 1,500 to $2,000 million. Nobody knows exactly what is the cost, because no company is going to really tell you how much money they have spent. Probably the exaggeration is done when they're talking about it so that they can recover more amount of money. But it is estimated that the amount of money required is to the order of about 1,500 to $1,800 million per drug. Now, while we do clinical trials, we require approval from two places. One is from the CDSCO. This is the office or the organization called the Central Drug Standards and Control Organization, which is headed by the Drugs Controller General of India. This is essential for initiating clinical trials of a drug. Approval is also required from the Institutional Ethics Committee of each organization where the trials are to be conducted. We do not conduct a trial at a single place, but usually the trial is done at anywhere between 10 to 15 different hospitals, so that you can complete the trial faster. You get the more patients by going to 10, 15 hospitals. At times I've seen trials being conducted over 15 different countries and 150 different hospitals. Such trials are also done. Now, in India, the applications to the drug controller at the CDSCO and to the ethics committee can be made simultaneously. You don't have to wait for one clearance to go to the other organization, but the trial can be initiated when both the approvals are in hand. So it doesn't matter whom you apply first, but only when you get both the approvals in hand, Only then you can begin the study. Lastly, there's a registration also required now. Every trial has to be registered on a registry, which is maintained by the Indian Council of Medical Research, and that website is www.ctri.nic.in. But this is just a registry, and registering a trial does not signify any approval from any authority. That has to be remembered. Now, the regulatory approvals, that when we make an application to the regulator in Form CT04, these are the documents that we will have to provide. They, as I said, clinical trial documents, informed consent, investigator's brochure, case record form, investigator CV, and the drug controller would grant the approval to conduct the clinical trials in Form CT05. Now, the question would be that suppose we wanted to come up with an alternative medicine or something like a traditional medicinal product for export. What method are we going to use? By and large, alternative medicine does not come under the CDSCO. It comes under Ayush. There's a Ministry of Ayurveda, Yoga, Unani, Siddha, and Homeopathy, and they handle all approvals or clinical trials for alternative medicines, whether they are for local sale or whether they are for export. Now, traditional medicines are also used through that way. People may think that are there any bottlenecks when we make an application? Yes, there are bottlenecks, but currently the government is working on trying to clear these bottlenecks. Earlier, it was quite common that when you make an application for a clinical trial, you may have to wait for a year, sometimes even more, to get the permission. Currently, the rule is that if the drug is developed in India, the drug controller will give you a permission within 30 days. And if the drug controller does not respond within 30 days, if he objects, that's a different issue. But if he doesn't respond within 30 days, you can assume that the drug controller has no objection and begin the trial. Now, for molecules which are developed abroad, this assumed approval is not yet allowed. However, there is a new bill that has come in which they have proposed that even for molecules developed abroad, a period of 90 days will be granted. And after 90 days, if the drug controller does not respond, you can assume it. But that bill has not yet been passed. So currently, you have to wait for any molecule which is developed abroad. You'll have to wait till you get the approval. Now, let's look at the drug discovery. As I showed you, the first you have the drug discovery, then you go to animal pharmacology, and then you go to animal toxicology. That means you're trying to find out in animal pharmacology, what does the drug do? What are the effects of the drug on the animals, which can be exploited? And then in toxicology, find out what are the safety issues. Is there any regulatory control over these things? Not really. The regulations do not speak much about how you should test the pharmacology of a drug. Actually, the pharmacology of drugs is so widely varying that no government can give us a guideline as to how the pharmacology should be studied. It all depends upon your own people, how the pharmacology is studied and how it is explored. But the drug controller has given guidance about toxicology. The drug controller does want to know what is the safety of the medicine. And for that, he wants to know the local safety, the safety after injection, the safety after oral administration, safety after single heavy doses, we call it acute toxicity, or a large number of doses is subacute toxicity, or a very long dosing period that is chronic toxicity. So what toxicity is required has been clearly defined by the drug controller, but the animal pharmacology has not been defined by any governmental authority, whether in India or abroad. The only permissions that you are required to take are something called from animal ethics committees, so that they check whether you are using animals logically and with proper care for the animals. Animals also should not be made to suffer unnecessarily. That is why Animal Ethics Committee is there to supervise the animal experimentation. Now, exactly how does one develop a drug, and how does one try to find out whether it has the particular activity or not? there are a number of methods now. Earliest, the method which we were using was blind screening, that any molecule that is synthesized or any molecule that is extracted from animals or from plants, we used to blindly screen it. We used to give it to animals and try to find out, does it have any activity? And with that, we would try to proceed to search out some molecule which has an activity. At that time, it was estimated that to find one good compound, you would have to synthesize and screen around 10,000 molecules. This was a rather wasteful procedure, and for years and years, laboratories worked on this without getting a single drug. Subsequently came computer-aided drug design, in which we do structure-based drug design and ligand-based drug design. So using the structures of the targets and the structures of the molecules, you try to find out what this molecule will do or what type of molecule you require to fit this particular target in the body. Currently, most of the work which has been done for COVID also is the repurposing of drugs. Now, this means that, see, most of the drugs that are in the market do not have a single activity. They have more than one activity. Some of those activities we may not even know them. Like hydroxyquinoline, which was an anti-malarial compound, was subsequently suggested to have anti-COVID activities. That it did not show very great anti-COVID activity is another issue. But that is called repurposing. A molecule which has been designed or developed for one particular activity is subsequently tried for a different activity. This is a faster method of drug development simply because you don't have to do any basic safety studies. They have already been done with those molecules. Now, repurposing is done by using two methods, again, usually data mining and computer-aided drug design. So these are the basic methods that we use. In the preclinical studies, as I said, that we are trying to establish safety and efficacy in non-human models. Why non-human models? Simply because about 95% of the DNA between animals and man matches. And whatever a drug does to animals, usually the drug does the same thing to human beings. Maybe at a different dose and maybe at a slight different kinetics, but more or less the same. Now, these studies can be done on isolated organs, which we can't do on human beings. You can isolate the intestine of a guinea pig and study the effect of the drug. You can isolate the heart of either a rat or a rabbit and study how the drug affects the heart. It's easy to study and the mechanism because you are studying an isolated organ without the influence of all other organs. You study it in isolation. Whole animals also you can use to do these studies. Now, how do they do that? Say, for example, if you take diabetes, we have drugs by which you can poison the pancreas of animals. So it produces a syndrome very similar to diabetes, but it is not diabetes. It's a model which is similar to diabetes. It's an animal model like the disease, but it is not the disease. Secondly, you can induce a diabetic disease in animal. So that is animal disease models are there. And finally, there are some human diseases that can be created in animals. And we do those things. And then we study the effects of drugs on that. Now, even though we may have studied the drug completely on animals, you cannot directly start using it in human beings. Simply because though there is a 95% homology between animals and man, the homology is not 100%. There are differences between animals and man. For this reason, will have to study the drug on human beings. And this we do in 3 phases usually. There is a phase 0 also, but we will take it up later. Think about it some other time. Generally, we do 3 phases, starting with phase 1, 2, and 3. Now, what is phase 1? Phase 1 is usually done to find out the safety of the drug. Hence, it is conducted in healthy human beings mostly. In some case of cancer studies, cancer drugs, we may use cancer patients, but usually we use healthy human beings. We use a small number of people, usually not exceeding about 50 to 80 people. And our main aim is to find out what is the effect of a drug on acute administration, a single dose administration, We may go into repeat dose administration or dose escalation studies. That if you go on increasing the dose, what is going to happen? If you go on repeating the dose, what is going to happen? And what is going to happen with a single dose? This is what we try to do in phase one. We also try to study the pharmacokinetics, that once you administer the drug to the animal, or to this, in this case, the human being, where does the drug go? How fast is it absorbed? How much is it absorbed? How does it get distributed in the blood and in the other parts, compartments of water? Then how is it metabolized and how is it excreted? We call it pharmacokinetics, ADME studies, absorption, distribution, metabolism, and excretion studies. After we are convinced that the drug is safe in the levels we expect a biological activity, then we go to phase two. In phase two, we pick those people who have the target disease for which the drug is meant. So if you're developing an antihypertensive drug, you will take patients with hypertension. However, we normally in this phase, we do not take patients with other comorbidity. So we don't want patients of hypertension to have diabetes also, to have thyroid problems also, to have cancer also, or to have some other problem. No, we don't want such people. We want people with purely one disorder, that is hypertension. Again, the number is not very large, rarely exceeding about 200 patients. And here what we are trying to see in the phase two, does the drug have the intended therapeutic activity? That's our main. And if it has, then we go to the phase three. The phase three studies are done on patients of the target disease, but these are the patients whom you are likely to see in the clinics and the hospitals. And we use a much larger number, sometimes very, very large numbers, 5,000, 10,000 patients could be used in phase three. Sometimes not so heavy numbers. It will all depend upon the incidence of the disease and all that. We'll come to those things also. And then after completing phase three, we make a request for permission for marketing. Phase three is usually done on a comparative basis, comparing it with a standard drug in the market. And then we try to see whether our drug is better than the standard drug. That is our aim. And then we come into the market, and then we would do phase four. The reason for doing phase four is that once the drug goes into the market, a much larger population of patients will be receiving the drug. Not only that, we are going to find people whom we had not selected for phase three being used in the market, being given the drug. So we might restrict our studies to patients between 18 and 60. But in phase four, patients much above 60 will also be using the drug. So we'll be getting a wider patient base and patients of the type which we see everywhere around us. So they're not very well selected or anything of that sort. And this gives a sort of a real world evidence. This is the real world. All the first three phases are more of an artificial conditions that we have created. The phase four studies are more real world studies. Now, what type of design do you use? At the present, there are many designs that we can use. The two basic types of designs are descriptive and analytical. And in analytical designs also, we have observational and interventional designs. But for drug discovery and for drug development, we mainly use the interventional drug designs. We do not use any of the descriptive or the observation studies in order to develop new drugs. Now, currently the gold standard for the design is as described here. First thing, the studies have to be prospective. We have a large number of studies which we do in a retrospective fashion. We take a patient who already has the disease, and then we go back into his history to find out what factors he was exposed to. Thus, we would try to know what are the factors that cause the disease. These are retrospective studies. Or we might take patients of a disease who have been given different drugs, and then we may try to find out which drug had the best activity. That would be a retrospective study. However, retrospective studies are quite biased, and they do not have the same sort of scientific acceptance. The most acceptable studies are prospective studies, in which we proceed from the present to the future. The trial that we want and the controller will accept are controlled studies, where you don't just give the new drug to one group of people. You have two groups of people where one group gets the new drug and one group gets the existing, usually the best available drug in the market. So that you've got to compare the effects of your drug with the effects of the other drug. The difference between the two groups will be that the two drugs which are used are different. There will be no other difference. So that whatever you see, whatever difference you see, can be attributed to the trial drug only. Now, we also want that all these studies should be randomized, because the human being is a very biased animal. We have our own biases on everything that we do. So what we want that participants should be allocated to trial groups in an unpredictable fashion. Neither the investigator nor anybody should be able to say which subject would go to which group. True randomization produces an equal sort of distribution of people. There is no way that two people can be equal, but two groups can be very, very similar. Our idea is to prepare or create two groups of patients, both which are similar to each other, and then try the drug on them. The next thing that we want is that neither the participants nor the investigators should know exactly which patient has received which drug, which patient has got the new drug, which patient has got the standard therapy. That should be kept blind. So that should not be revealed. Now, this reduces the bias both of the patients as well as the investigators. The patients have a tremendous bias. All of us have bias. There are some people who love new drugs. You give them anything new and they're extremely happy with it. And that happiness translates into better cure rates. Similarly, investigators are there that most of the investigators have a feeling that whatever they investigate should succeed. We all have that feeling. And that is why the investigators must be kept blind. Otherwise, the bias of the investigators also comes in. Currently, the gold standard for clinical trial design is a prospective, controlled, randomized, and double-blind study. And that is what we say that is the best design currently. There are other types of designs. I'm not saying that they're bad, but this is the best design, which may not be always possible. There will be situations in which you are unable to do a double-blind. There will be situations in which you can't do a prospective study. There will be so many problems, ethical, legal, so many problems coming. But this is the best design. If it is not possible, then we'll go to a slightly lower design. Now, how do you select the site? The sites are selected by the sponsors as per their choice, usually depending upon the prevalence of the disease and the availability of the patients. Now, if you want, say, patients of tuberculosis, and you go to Jaslok Hospital or Leelavati Hospital, you're rarely going to find a TB patient there. For that, you have to go to JJ Hospital. There you will find the patient. So the sponsor should know what type of patients are available in which hospitals. Secondly, the site should have adequate infrastructure, well-trained staff, qualified and well-trained staff, experienced investigators to do the trial. Every site signs a tripartite agreement. This agreement is between the sponsor, the investigator, and the site. And all the three parties are responsible for taking the trial to its fruition. The site is responsible for providing facilities and staff and for the participants' well-being in case the investigator suddenly leaves the trial. Many of the investigators are old. They might die in the middle of the trial. So what happens to the patients who are in the trial? The site must ensure that another investigator takes over the patients and continues the trial. Now, how do you choose the subjects? Subjects are chosen on two bases. We got two types of criteria, inclusion criteria and exclusion criteria. First, to the entire population, you apply the inclusion criteria. Inclusion criteria first selects a small subgroup of the population. Okay. We have applied the inclusion criteria. Inclusion criteria usually include age, gender, diagnosis of the disease, and willingness to participate. Like we would say that we want patients between the age of 18 to 40 of either sex, all having a type 2 diabetes, say moderate diabetes, and those who are willing to participate. So this is the inclusion criteria that we normally define. In most cases, we choose the age group of 18 to 60, though you might have patients at 95 also. There would be a 95-year-old who comes for the treatment. True, but we don't take that person in trials because the chance of that patient dying is extremely high. And we do not want patients to die in our trials because then we'll have a lot of administrative problems. We'll have to prove that the death was not related to the drug. There'll be a lot of issues in it. Now, to this selected subset, we apply the exclusion criteria. The first exclusion criteria is pregnant and lactating patients. We don't want pregnant or lactating women in the trial because when we give a drug, we are not affecting one individual, we are affecting two individuals. Without taking the permission of the fetus is going to get the drug. And many times we do not know what will be the effects of the drug on the patients. Most of the drugs are metabolized in the liver and they're excreted through the kidney. So any patient who has liver or kidney dysfunction will not suit our purposes because the kinetics of the drug will be different in such patients. Certain other diseases might increase the risk of the patient. That's why we remove those people. Like you see, if you are talking about an infection, if you are talking about the test, you are going to do a trial of an antibiotic. We'll take people with infection, but we'll remove people with diabetes because diabetic infections are very difficult to treat. So they are going to complicate your results. So we remove them. We also remove patients with psychiatric disorder and those who are in substance abuse because they're not dependable. You tell them to take one tablet twice a day. You can't be sure whether they will take. Then lastly, we'll remove those patients who are taking drugs that might interfere with the metabolism of or excretion of the new drug. There's something like grapefruit juice. Grapefruit juice, which is not very common in India. This grapefruit juice interferes with the metabolism of many drugs. That's why this very often we try to find out what all things does the patient take. If a patient is taking cheese, it affects the metabolism of certain psychiatric drugs. So that becomes a problem. So there we'll have to look at it, what the patient. So these are the general exclusion criteria. There will be other exclusion criteria like patients taking steroids, patients with who are receiving radiation. These patients might react differently. So how do you select the patients? From the total population, on the total population, you apply the inclusion criteria, you get a subset. On this subset, you apply the exclusion criteria, you remove those patients who are at risk, and then you get the population which has passed through. That is the population that you will take. Now, after you have done all this, you must get the permission of the ethics committee. Now, every hospital where you do the clinical trial would usually have its own ethics committee, and you have to take their permission. You will have to submit all the documents that they want in the format that they want, and then they will hold a meeting. They'll discuss your documents. You may have to explain the trial to them, and then they'll give you a permission. Now, the ethics committee has to be constituted as per chapter 3 of the new drug and clinical trial rules, and ethics committee members must have the requisite qualifications. They should be trained in clinical research, trial regulations, guidelines, and they must also undergo periodic training. Now, it may happen sometimes that the site where you want to do the trial does not have an ethics committee, or an individual investigator wants to do the trial and he doesn't have an affiliated ethics committee, in which case you can go to another ethics committee which is within 50 kilometers range, a radius of 50 kilometers, and get their permission. However, remember one thing. If your own institute has an ethics committee, then you cannot go to another ethics committee. Then you have to go to your own ethics committee. So you can't bypass your ethics committee and go to another committee, right? Now, the IEC approval, again, the IEC is going to look at all these documents and they're going to look at it very carefully. They will ask for 21 days to study these documents. They'll go through all the documents. They'll also look at something like insurance details. Because if a patient suffers during the trial, some injury, some serious illness, then the patient has to be provided free medical support. And if the injury is permanent, the patient may have to be given a compensation. All those things come from the insurance. That is why the ethics committee is going to ask you a lot of questions about from where are you going to raise the money? Where do you have the money to pay all these things? IEC will provide the approval if all ethical conditions are met. And we'll briefly talk about the ethical conditions also, what conditions they are. And that is how you can begin the trial. Now, this IEC, its role is to protect the rights and well-being of trial subjects. And IEC must uphold the principles, the three main principles of ethics, that is respect for persons, beneficence, and justice. IEC also works according to written and approved standard operating procedures. IEC has the authority to approve your study, to demand modifications, or to reject the proposal. Rejection is rare, but demanding modifications is quite common, and acceptance is also quite common, but sometimes the IEC may reject. Throughout the trial, IEC will do periodic analysis of what you are doing and will demand periodic reports from you. And should a serious adverse event take place, should a subject suffer a serious adverse event, then the IEC will analyze that event and decide upon medical reimbursement to that individual and the need for compensation. Now, we will briefly talk about the three principles of ethics. The first principle of ethics is respect. That research participants are to be considered as autonomous. Each individual is autonomous, not only research participant, you and me, we are all autonomous. That means we have a right to decide what we should do or what should be done to us, right? And no person can be taken into a trial without his voluntary consent or enrollment. Now, some people lack either the legal or the medical capacity to take such decisions. A person may be mentally unsound. Such a person cannot take a legal decision whether to take part in the study. That person is known as vulnerable. Any individual below the age of 18 does not have a right to take such decision. That individual is also vulnerable. The government has defined who are vulnerable, and such people the ethics committee will be very, very careful to ensure that their rights are protected. They will be provided with additional protection. But they need to be included in trials if the drugs may benefit them. Some people would say that, why do you want to include babies and children in clinical trials? Of course, we need to do it. Otherwise, how would we know that the drug is safe for children and babies? And tomorrow when a baby falls sick, what drug will you give? If you are not used them for testing, you won't know which drug is safe for them. That's why the trials must be done on them. The second principle is that of beneficence. Everything that an investigator or a sponsor does must ensure benefit to the participants, to the community or the society. But remember, societal benefits do not justify harm to participants. You can't say that, okay, this participant may die, but society will benefit. No, that's not permissible. Now, expected benefits from any study must always be more than the risk to the patients. So there has to be a balance between risk and benefits, which we always see in life also. Here also you have to. Unfortunately, both these things are not easily quantifiable. So you'll have to put in a lot of effort to find out whether the risks are okay enough, have they been minimized or not. Also remember that it is the duty of the investigators to maximize the benefits and minimize the harms. Whatever you do, reduce the harms, increase the benefits. And any trial in which the participant may be harmed or may die is unethical. You can't take up such a trial. So this is the principle of benefits. The third principle is the principle of justice. All men are born equal. And when we say men, we also mean women. There shall be no difference between them. Now, risks and benefits and opportunities should be equally and fairly distributed. It cannot happen that you give risk to one group and benefits to another group, which has been often said that clinical trials are done in India so that our people take the risks, and when the drug is to to be marketed, it's marketed in America, so they get the benefits. Of course, this is nonsense. It doesn't happen. If you look at the number of trials which are done in the country, you will realize that this is not true. But this is the comment which is often made. There should be no discrimination on between participants on the basis of gender, age, religion, caste, color, social or financial status, or any artificial barrier should not come in the way, and treatment should be equal. But remember, equal treatment is not really justice. The principle of justice is far more complex. The principle of justice is more complex, and we'll have to study it in greater details. Now, the clinical trial phases, which I said, the 1, 2, 3 phases, we'll briefly again revisit them. They're usually conducted in phases. Phase 0 is rarely conducted in India and is rarely required for most of the drugs. But before each phase is initiated, regulatory and ethical permission is essential. Phase 1 is not usually allowed on drugs developed abroad. So if a drug is developed in America, we tell them that you do the first in human study, you do it in America, and then you come to India, then we'll allow it. And each phase has an increasing number of subjects, and each has its own aims and this one. So phase 0 was introduced by the FDA about 15 years back, 15, 16 years after 2006, where you use a very tiny dose, 1/100 of the expected calculated dose. We use it. Usually the ceiling dose is 100 microgram. Our main aim is to study the pharmacokinetics, where does the drug goes and what it is likely to do. This has not been accepted by all, but one thing is that when you use such a small dose of the drug, you require highly sophisticated instrument to detect, and it is a very expensive job. Phase 1, as I said, is the first time in human study. It focuses on the safety of the drug. Small number, maximum 80 patients you may use, usually healthy people. You may do a single ascending dose. So to one person you give 10 microgram, to another person 20, then 40, then 80, increasing doses, or you do multiple dose studies, depending upon the ultimate use of the drug, proposed usage of the drug. It's always conducted in specialized units like ICUs by clinical pharmacologists and domain experts. Phase 2 studies are conducted by experienced physicians and pharmacologists on patients who have the target disease. Usually the number of subjects is anywhere between 80 to 200, and relatively homogeneous population with low incidence of comorbidities. This study may be double-blind, positive controlled or placebo-controlled, and multi-centered. And on an average, about 30% of the drugs advance from this point to the phase 3 study. Now, phase 2 study is also, the aim is to study the efficacy of the drug in subjects with the target disease. We also study safety, side effects as usual. It may be double-blind, positive controlled, placebo-controlled. Sometimes we use a phase 2A, phase 2B. Sometimes we split these phases depending upon our needs. The phase 3 is the penultimate stage, or you can say pre-marketing. It is the ultimate study conducted on large number of subjects who have the target disease to demonstrate the safety and efficacy of the new drug in comparison to the best available in the market. These are usually prospective, double-blind, controlled, randomized, and multi-center study. And study population must resemble the end users. So this is actually the crucial study. Usually we would do maybe 2 or 3 crucial studies, and the size will depend upon a variety of factors, usually statistical power, the guidance from the DCGI, and how many people are expected to use it. Oral contraceptives were tested on millions of women, okay, in the 60s. Now it's quite common to use thousands of patients for this phase. It's often conducted on outpatients, ambulatory and working populations. Comparison is usually with standard therapy. It's rarely with placebo. Please forget the placebos. Most of the trials which are conducted now are always compared with standard drugs, rarely placebo. There's very special conditions. And these trials may be multi-center studies done on in different regions with varied ethnic groups and all. Again, phase 3, we often split it into phase 3A and phase 3B. I'll give you a typical example of an antibiotic. which can have many uses. So what we would do is that the most common infections in human beings are respiratory tract infections and urinary tract infections. So we begin the studies on respiratory tract and urinary tract. Along with that, we'll begin studies on infections like osteomyelitis, on say meningitis and ophthalmic infections and abdominal infections. We might begin all the studies. But the first ones to be completed would be the respiratory and urinary studies, because you get large number of patients. So we usually split that into 3A and 3B. So 3A are those studies which are going to get completed first. So the minute we complete those studies, we go for the permission for marketing. Now, while we get the permission for marketing, we try to complete the 3B study, the additional indications like meningitis and skin infections and other less common infections, so that hopefully we will be able to push our antibiotic into the market as early as possible. As soon as the phase 3A study is reviewed, we go into the market. And then maybe after 2 years, 3B gets completed and we'll take additional indications for that. Now, phase 4 will be always conducted after marketing of the new drug. But why are we conducting it? Basically for additional indications. Does the drug have any other advantages? Can we exploit that? What about different subject groups? Usually, as I said, we do not use pregnant women or lactating women. We try to find out, can it be used safely in that population also? Or can it be used safely in children? Can it be used safely in geriatrics? Can we change the dosage schedules? Can we bring down the dosage? Is it still equally effective? And do we find more adverse effects? You see, some rare adverse effects might be found only in the phase 4 study. They're not seen in phase 3 studies, because phase 3 studies have limited number of people. As you go on increasing the number, the rare effects are seen. So that's why we do the phase 4 studies. And then remember, from the marketing point also, as our drug becomes older in the market, newer drugs keep coming. So the company must know where your drug stands in the presence of all the other drugs in the market. That's why you continually do the phase 4 study, find out where your drug is, how good is it? Is it better than the newer drugs coming in, or is it also losing the market? If it is losing the market, then you will have to start developing another drug to that, because this bread earner of yours is going to go down if a better drug comes to the market. So you must have another drug to fall back in. Now, there's a lot of freedom in deciding the trial design. Sometimes observational and interventional trials are permitted in this phase 4. Here you may try different designs, but not in phase 3. In phase 3, the situation is strict. Now, remember one thing, that most drugs have adverse effects. Most drugs, including aspirin. And if you talk about anti-cancer, the adverse effects can be very, very serious. So we define adverse events as anything that unwanted happening to the patient. Now, some of them are considered as serious adverse. There are actually 5 categories: those that lead to death, which are life-threatening, which lead to hospitalization, or which produce permanent injury, or lastly, which produce damage to a fetus in the in utero. So these 5 types of adverse events are known as serious adverse events. And whenever such a serious adverse event occurs, the sponsor is required to reimburse the cost of treatment. And in case of permanent injury, the compensation has to be paid. Now, the compensation is given on the basis of a mathematical formula which has been derived in 2013. The lowest compensation for a death which is related to the clinical trials is 2 lakhs. And the highest compensation is rupees 75 lakhs. Now, the formula was first developed in 2013. Now, the formula needs to be revised because the value of money has gone down, the interest rates have changed, inflation has changed everything. That's why adverse events take place. Now, to look at the world, what is happening in the world, how many trials are we doing? There was a time when the figure for India was only about 1,000. That's not too far, about a decade back. There were just 1,000 studies that India was doing. Now we are doing a pretty respectable number. We have beaten Russia. We are at 8,000. We are close to beating Maybe next year we'll beat Australia. South America is not too far, 13,000, we are 8,000. And trials in India are growing very fast. During the COVID pandemic and post-COVID pandemic, clinical trials have seen a great increase, a large increase in India. One question, of course, many would ask you, because some of you are going to be innovators, some of you are going to develop new drugs, and there would be some people who are going to say that, can I enter the drug development industry, clinical trial industry? And they would like to know what is their future in this industry, what is the scope that they have for the professionals, what scope do they have? Now, the scope a person in clinical research has will depend upon the scope of clinical research itself. If clinical research keeps rising, then the scope for people will keep rising. We have looked at what are the trials which are taking place and how are they increasing right from 2014 to 2022, and the projections for 2023, 24, 25 are there. As you can see, there's a steady rise, and actuality, the CAGR is increasing. We are growing at a CAGR of anywhere between 11% to 12%. And in the year 21 and 22, we have shown a much higher growth. Actually, this graph tells us what was expected, but the actual figures are slightly better than what were expected. And with this slide, I come to the end of my presentation. As I said in the beginning, normally this entire process we cover in 10 to 12 hours. Today, I've tried to cover it in 1 hour. So I have been able to only scratch the surface. And that's why I've just called it an overview of that. I'm sure you have questions, and if you have questions, put them in the QA box. As Dr. Pranita said, that you can raise your hands. We'll be happy to answer them. I have given my email ID, and you may write to me. And if you have any question, I'll be happy to answer. So thank you very much for your patient hearing. Now, Dr. Pranita, I hand it back to you.
Yeah, thank you. Thank you so much, sir. It was really, really informative. And we have two questions in the chat box, and I request participants as well, if they have any questions, um, uh, you can raise your hand, and we can unmute you so you can ask your question on your own. Uh, I think we have one question which is relevant to what I mean the graph which you showed, sir, towards the end. What it's from Suchita Sen. What percentage of drugs move to?
May I interrupt, Dr. Pranita? Can we
Sure.
start from the top? Because I think Nidhi Mehrotra's question has come in much earlier.
Oh, sure, sure.
So if you don't,
Oh, sure, no
I thought
problem.
that we'll give them. Okay, Nidhi
Okay.
Mehrotra, so you
Uh,
said that post-trial access is not mandatory by the law as is in case of Brazil. No, it is mandatory in India. Rule 27 of the new drugs and clinical trial rules have specified that post-trial access is mandatory. Only thing that it has to be recommended by the investigator and it has to be approved by the ethics committee. But I, as an ethics committee member, accept that post-trial access is mandatory.
okay, sure, sir. I think,
That is possible.
yeah.
The anonymous attendee, in clinical trials, a drug is URI. URI means is URI, but have you observed another better activity? For example, anti-inflammatory activity of the same drug that has been explored for the. Yes, of course, we have so many examples of this. If you take aspirin, the simplest one, aspirin came in as a painkiller and an anti for fever. Subsequently, we came to know that aspirin inhibits platelet aggregation, prevention of coagulation of blood. Subsequently, we have come to know that aspirin prevents premature births. The trials have been completed. Currently, trials of aspirin are on for the prevention of recurrence of cancer. So many new applications of aspirin have come in. And why I took aspirin? Because today aspirin is 125-year-old drug. It came into the market in 1897. It's now 125 years that it has been marketed, and still we are getting new uses of aspirin. So we have seen this. What percentage of drugs move to phase 2? From phase 1 to phase 2, it's anywhere between 60% to 70%. But Suchita, let me tell you one thing, that these percentages really do not mean much, because if your drug doesn't make it to phase 2, for you, it means nothing. No? There's no satisfaction that 70% of the drugs move to phase 2, but your drug failed. So it really doesn't make any sense. And secondly, this percentages that so many percent go here, so many percent go there, they don't help us to predict what will happen to an individual drug. It is just an analysis, it's a post-mortem after the drug has come into the market or it has been thrown out. It's just a post-mortem of that. Which is the certifying body? No, the drug controller. Every time when you want to move from one phase to the second phase, you make an application to the Drug Controller General of India. If the DCGI agrees, he gives you a permission. If he doesn't agree, he says no. So the certifying body is this one. For medical devices, any clinical trials, studies are different. Yes, there is a difference, a slight difference. First thing, we don't do 3 phases. There are 2 phases. They're called pilot and pivotal phases. And there is a slight change in rules here and there, the names of the document changes. There's a difference. The reason is, you see, drugs, whether it is aspirin or it could be an anti-cancer drug like trastuzumab, the risks are the same. And we have to be very careful with all the drugs. I would say that with drugs like aspirin, we have to be more careful because a very, very large population is going to use that drug. Just a minute, please, there's some noise. I told him it is going to 75. There's some repairs going on in our building, so that's a noise. Anyway, so for medical devices, there are differences. Some devices, for example, like a tongue cleaner, for example, or a tongue depressor, you don't require any clinical trials. And for other devices like a stent, you'll have to do very serious clinical trials. So the treatment of devices is quite different than the treatment of drugs. So there is a difference, but the rules for devices are different from the drugs. And that's why we did not mix drugs and devices in one lecture. It would be more difficult for me to explain it, and it would be more difficult for you to understand it. Do nutraceuticals have any regulation? Yes, of course. See, nutraceuticals, there are new rules which have come in, and there are regulations for nutraceuticals. You can go to Google and you can download the rules for nutraceuticals, or you can send me your email ID. I will send you the regulations for that. There's a full set of regulations for that. If some SAE occurs in a case of a surgical device, the formula that we use for compensation calculation is standardized. Currently, we are using the same formula for compensation. Whether it is for a drug or a device, we use the same formula. Does DCGI approve preclinical trials done at any recognized animals? See, every laboratory where you do animal studies must have what is known as GLP certificate, Good Laboratory Practices Certificate. And only those laboratories you can do the animal studies, preclinical studies. That is the requirement. Is there any way through government agency to see if your invention can go through a clinical trial? No, you make an application. Generally, if you go to a pharmacologist, like I'm a consultant, if you come to me and you ask me that, do you think that this application will go through the clinical trial? I might be able to give you a good answer and advice that whether there are good chances it will go through or there are not. that the chances aren't there. Then I would leave it to you that you want to try it. Suppose I tell you that, look, there is no chance that the government will approve. Yet you feel that you want to make an application, you're free to make an application. Most probably our uh, prediction will turn out to be true. But if it is not true, it's your good luck. Are off-label indications used for phase 3B? No. Phase 3A and 3B doesn't talk about off-label indications. Off-label indications are those which are not included in phase 3A or 3B. Which are not in that are known as off-label indications. I would, the process same for drugs applied topically. Yes, the more or less the process is the same. The trials would be very similar. They'll be less complicated because the drug is not going to be absorbed systemically to the same extent. So you're going to have a slightly easier way through the regulations. Bipin says, how are drugs dispensed to patients in phase 3 trials when the sample size is large? No, usually we make bottles of tablets or strips of tablets of the required number, and we distribute them. That's a totally different science. And usually there are companies which do that for us. So we tell the company that we want the product packed in, say, in 29 tablets per bottle or 30 tablets a bottle, whatever we want. And then the bottles are dispensed. We have designed a trial for a organization where we have to give 22 vials per patient. So we have created a separate box. Those 22 vials are packed in that, and one pack is given to the patient. So different ideas are used for different dosages and different numbers. Drug effects depend upon person's immunity in clinical trials. Whether person's age, see, all drugs are not affected by immunity. There are few drugs that are affected by the immunity and age and all that. But by and large, we are choosing the age between, say, 18 to 60, where more or less the immunity and all, they're more or less similar. So the differences are not many. But in human beings, the differences between two individuals are significant. And that's why we need a large number of human beings to find out what is the effect of the drug. If you are doing an animal study, 6 animals is enough in one group. But in case of human studies, statistics will tell us that sometimes we need to have 100, 200 people per group. This is because of the innate differences between individuals in human studies. Vaishali,
Sir, I think we have missed 2 questions by Bipin. Yeah, he was asking, are patients expected to maintain the record of drug consumption and submit the same?
No. Patients are not expected to maintain the record. What we do is that, suppose we are calling the patient back every 1 month for a periodic check. Then we give the drug only for 33 days, and we ask the patient to return the unused tablets. From that, we calculate what is his compliance. That is the way we don't ask them to maintain a record, but we try to find out whether he has followed the thing. How do clinical trial companies collect this data? This is the way. You see, have they returned the right number of tablets? And we don't tell them that why we are asking you the tablet back. We never tell them that. So they just return the unused tablets, and from that we calculate what is the compliance. Now it can happen that somebody is non-compliant, and that does mess up our study. That's why people who are not reliable, we usually don't take them in the trials. Is that okay? Drug effect depends. Okay, immunity we say all yeah, fine. If the drug is already approved, and in the same drug, if we have made slight modification like conjugation, we need to go through all the 3 phases. You make the slightest modification at any position, the drug is different. So the drug is not the same, the drug has changed. So it becomes a new drug altogether. Is DCGI aware of different animal models for different cancers, or do we have to get specific animal model approved first? See, it's not a question of DCGI being aware. Every time when you use an animal model, it is up to us to demonstrate how good the model is. And remember, no animal model is perfect. How good the animal model is. That is why we use more than one model. We don't depend only on a single model. We have to demonstrate and we have to give references to say that I'm using this model. This model has been used by these papers, and this is what is seen. And standard drugs produce these effects on this model also. So you use standard drugs to show that your model is equivalent to the earlier models also. Is that clear? Can you elaborate in the next session? Oh yeah, surely if you want, we'll certainly have a session on medical devices. I have no problem with that. Madhuri Shinde, it's the same drug with another novel property. Still, we need to go through phase 1. No, we don't need. If it is the same drug with another property, we don't need to go through phase 1. That is why it becomes faster. We don't need to go through the laboratory studies, the animal studies, and phase 1 is not required. When the drug is the same, we require only phase 2 and phase 3. That's why it is faster. Smitha, do medical device for external testing, say saliva testing require? Of course, they do require approvals. Yes. Saliva testing or whether it is urinary testing, they do require approvals. Like, for example, the pregnancy test. Now, suppose you start getting unapproved pregnancy kits in the market. Do you realize what havoc they will cause? Every kit must have what do you say, it should be tested and it should have results which are acceptable. Approvals are necessary for even whether even if they are saliva or urinary testing ones. Everything requires it. How much do the regulatory consultants charge for advising or guiding through the medical device approval process? It depends upon the consultant. You have consultants who will charge you 1 crore, you have a consultant who will charge you lakhs. I can't say that how much will a consultant charge. You'll have to talk to individuals. Like if you talk to CROs, we've got the world's biggest CRO is IQVIA. For any trial, their charges go in crores. But you do it yourself. It might cost you maybe 1 lakh. That difference will always be there. It's nice inside to work in. It's very useful. We have wound healing film, which is prepared from natural polymer from sea waste. Shall we go in for trial and licensing to CDCO or Ayush for? Now, whether you should go through, you see, if you go through Ayush, it will be faster. I can tell you that. But in the market, a product which has come from the Ayush stable will not carry that much respect. Please remember that. So what do you want? You want to go through Ayush? You can go through Ayush. You want to go through the modern medicine? It will take time. It will be more expensive. But when it comes to the market, people trust it. Can GLP lab do clinical? Oh, no. Clinical trial cannot be done in a GLP lab. GLP lab does animal studies. And clinical trial means human study. Human study cannot be done in a GLP lab. Sorry. Can you please elaborate the use of AI and ML in clinical research? My God, somebody from IIT Bombay asking me this question is going to be very difficult for me to answer. Well, you see, I'm not an expert on AI or ML. I know people who are using AI and ML for these things. I would suggest you go to the proper expert for that. I'm not the expert. Could you please share your email or ID? Yes, I think I shared it. It is ravindra.ghui@gmail.com. I will try to share it again. Yeah, I hope you can see it now.
Sir, I'll put it in the chat box.
Yeah, please.
Yeah.
The question answer session. Now, excellent session, well structured. For clinical trials that don't have a drug, but are based off biopsychological therapeutic models, how do we go about addressing AEs and AEs? You see, if you're using an intervention, sometimes a psychological intervention also, you can cause a lot of adverse effects on the individual. So adverse effects and SAEs are treated in the same way or a similar way whenever there is an intervention. Now, your intervention could be a drug. It could be a device like a bandage. Suppose you see a wound healing plaster. That could have an adverse effect. So that will also be treated more or less in the same way. Are patients expected to maintain records of drug consumption? As I said, no, they are not expected to. We calculate that data. Can you elaborate the role of insurance in clinical trials? You see, currently insurance is not compulsory. The sponsor is supposed to cover the expenses for medical reimbursement and compensation. From where the sponsor gets that money, it is the headache of the sponsor. Insurance is one good way of getting that money, but if you don't want insurance, you need not take insurance. If a company failed in phase 3, then the company has to start from phase 1 again. No, you see, if a drug has failed in phase 3, you cannot start that drug in phase 1 again. It is out there. Once you have failed in a phase 3 study, that drug doesn't get tested again at all. The company doesn't fail. It is a drug that fails. In case when one is studying effects of existing drugs or interventions, how do you go about with the ethics process? I'm not clear. Sachin, if you could write to me this question, I'll tackle it separately. I'm not clear with the question. Sachin Ganokar, please. Ganorkar. Few drugs effect we get on body shows off after a few weeks or months. How much is mandatory? You know, it really doesn't happen. Though this talk is there that a drug given now may produce its effect after 1 month. No, it doesn't do that. And there is no effect that I've never heard of a drug which produces an effect after a week or 2 weeks and doesn't produce an effect immediately. Do let me know if such a drug exists. We'll study it again. No problem. Has there been any neuro-related drug development in India? Oh, plenty. Just go to CTRI, you'll find a lot of drugs which are being clinically tried in India. There are neuro drugs. There was recently a drug for neuropathy which had been developed by Glenmark. It is currently under trials. So drugs are there. Thank you very much, SINE, Dr. Pranita. Are there 2A, 2B studies as well? If yes, these are parts of the phase 2 studies, Abhijit. Parts of phase 2, which can be often split in 1 or 2 parts. The reason for splitting these is different, that you might want to study 2 different effects. So you take it 2A, 2B and do that. Shiv Kumar, thank you very much, Dr. Very informative. I think that makes it. If there's any other question, we can handle that.
I think, sir, we have, I think, 2 hand raised. I'll unmute them so that they can ask
Sure.
you the question.
Please.
I think Smita, I have unmuted you. Could you please, can you please unmute yourself and ask question? Yeah.
Hello, Dr. Ravindra.
Hello.
I had asked you a question about medical devices for saliva. So how different are the medical device regulatory for external testing versus internal? So there could be a medical device for testing blood sugar.
In medical devices, there is a special clause called in vitro diagnostics, where the device is not introduced into the body, but outside the body, it is used to diagnose a disease. Like you might use saliva, you might use urine, you might use sweat, you might use any body fluid, or even say a thermometer. You're using it outside the body, you're measuring the temperature. So all in vitro diagnostic devices have got a particular method for studying them. And if you would like to refer to it, it is called the Medical Device Rules of 2017. They're freely available. They're on the CDSCO website also. You can Google them. You'll get the Medical Device Rules 2017. You'll be able to download the rules.
They have too many jargons.
Of course, they're rules. They're made by bureaucrats. There will be jargon.
Yeah, the language is very odd.
Of course, that is. If you look at the drug language also, it is odd. But that's our job to understand that language. So we have to do it.
Yeah, thank you.
Welcome.
Subhashini, could you please unmute yourself and ask your question? Subhashini, I feel there is some technical issue, sir. However, I have one question, sir. Uh, I mean, nowadays we heard a lot of platforms or in silico
Yes.
screening of the drugs. Then there are lab on chips kind of a method for screening and different probably phases which can be used for different phases. I mean, I would like you to please comment on that and how could that be related, can be used in future.
You see, we have a lot of in silico methods, and these depend upon when for particular diseases when we have identified the targets. Usually, the target for every disease is a biomolecule. It's a large biological molecule which has certain active sites, and the rest of it is inactive. Now, the whole idea is that if you have identified the active site, and if you want to block the site, by looking at the site and studying its chemistry, their bonding structures, electronic bonds around that site, you will get an idea that what sort of molecule is likely to go and block. You won't get the final answer that this is the molecule that will do the job, but you might get about 5, 6 different molecule options that these might do the jobs. So instead of trying to synthesize 10,000 molecules and then check them, you synthesize only 5 or 6 molecules and then you check them. So basically, this is the theory with which it works. We have certain companies in Pune also. We have got a company called Nova Leads, which works basically on this mechanism. So their what you say motto is to develop drugs using this platform. Secondly, what they're doing, they look at existing drugs and they looked at the possible targets for other diseases, and then they think that can this existing drug work for another disease? So that can be done by deep mining, it can be done by computer aided drug design, but then the sort of expertise which you require for that are people who are from machine learning and AI systems. I'm not from that system, so I can just give you a general idea about it. Certainly, these methods cannot be used in place of clinical trials. Currently, clinical trials cannot be replaced. And even animal studies, though they are thinking of replacing animal studies by purely in vitro studies, there is work going on. Some people say it is possible, but as yet, animal studies have not been replaced. Currently, what we do is to the 3 R's: refine, replace, and reduce. Reduce the use of animals, replace animals as much as possible by other methods, and refine your. That is what we are doing. And one day we hope that with all the AI and ML available, we will be able to predict and bring out a molecule that will directly go into clinical testing. But if that happens, remember in 2006 what happened? One drug had gone through extensive animal testing. It had proved to be absolutely safe. But when it was injected in human beings, it produced a cytokine storm, and 4 out of 6 people ended up with multi-organ failure, and damages worth millions of dollars had to be paid. So people are still worried. Okay, AI is good, ML is good, but can you rule out the possibility of such an accident? That is the worry.
Got it, sir. Thank you
Yeah.
so much for that. I think Subhashini is able, I think she has
Back
unmuted
on.
herself.
Yeah.
Subhashini, would you like to go ahead and ask your question? Uh, I still feel there is some technical issue. Subhashini, you can write to us and we can try to get your question addressed. I think with this, we are almost towards the end of the session, and we have covered all the questions which are there in the Q&A chat box as well. If there are any more questions, please do write to us as well. You can write to us at SINE at sineIITB.org. I'm putting our email address in the chat box as well. So if there are any more questions, you can forward it to us as well. We'll try to get it answered, and we'll try to arrange another session for medical devices as requested by the one of the participant here. And we'll try to see that most of your questions on medical devices side are also addressed. So with this, I would like to thank Dr. Ravindra Gui for his valuable time and such an informative session. So even I would like to, I got to learn more about clinical studies for drugs. And this is, I think, one of our first session on drug side. Which we have conducted. So far, we have conducted sessions on devices, I think genomics and certification and other area. This is our first session on drugs, and we are really thankful for you, Dr. Ravindra Gui, for this information and guiding us through this. And I also like to thank all the participants here for attending these sessions and even participating in very active Q&A here. And thank you for posting questions. And we would like to see you in our next session as well, which we are conducting on 12th May, where we are going to conduct on the devices side, but we are going to cover the approval, FDA approval, overview of the FDA approval for the devices. So we hope you attend that session as well. And with Dr. Ravindra Gui's permission, probably will upload this session on the YouTube channel as well, where you can access the recording of this session. And if you would like to know more about that, you let us know. So thank you so much. Thank you, sir, and thank you, everyone.
Thank
And
you.
have a great weekend. Thank you.
Thank you, Dr. Pranita. I would like to thank the organizers of SINE and all the people who have attended, heard me so patiently, and shown the interest. Thank you very much.
Thank you.
Have a good day.
Thank you.